In this video, we're going to be focusing on cellular communication, or how cells receive information about the outside world in various forms and then respond appropriately. And this is a process that's fundamental for life. Obviously, the environment outside of a cell is constantly changing, and cells need to be able to get information about their cellular environment, their cellular context, in order to be able to adapt their own internal environment appropriately as a result. And as you can well imagine, in the context of a multicellular organism, it's really important for the various functions that are associated with different tissues and organ systems in the body to be coordinated with one another. And cellular signaling and long-range communication between different organ systems is one way that that can be accomplished. In this video, we're going to go into a fair amount of detail about some example signaling pathways that are representative of the most common types of intracellular signal transduction pathways that exist out there. But to better frame the discussion, we're really going to want to keep in mind some fundamentals of what we want to consider when thinking about cellular communication. So first of all, just understanding why it's important for different types of cells to be able to send and receive signals from the outside world. And all cells do this, and depending on which organism you're looking at, the context is going to be different, whether you're talking about a bacterium being able to sense nutrient levels or accumulation of wastes and toxins and pH in its environment. That's going to be very important for that cell. But like I said, in the context of a multicellular organism, it gets quite a bit more complex and is not just a function of a cell receiving information about its immediate environment in order to be able to respond, but also to be able to coordinate the activities and functions of various organ systems so that they're all sort of synced, right? So that's one thing that we want to understand. Why is it important for cells to send and receive signals? Secondly, what is the nature of the signal? So cells can respond to a whole host of stimuli, and in this discussion we want to get a sense of the diversity of those cellular signals that can be responded to. Thirdly, what are the main types of receptors that cells use to detect the chemical signals or other signals? Because as we'll see, there are other signals in addition to chemicals. And then what happens inside the cell as a response to that stimulus to affect some kind of cellular response. And what we're going to see is that there are very often molecules called second messengers that are going to be involved with that intracellular signal transduction process that are going to be able to diffuse away from the cell surface and interact with other molecules inside the cell and affect their behavior and their function in order to elicit some specific cellular response. Then we'll want to focus on what types of cellular responses are possible. So in the cytoplasm, in the nucleus, we could be talking about enzyme activation or inhibition, or we could be talking about changes in gene expression. So we'll take a look at some of those specific responses. And then finally, how do cells terminate the signal? In order to maintain responsiveness, obviously they have to be able to respond to a signal and then stop responding so that they can be re-stimulated as appropriate. All right, so we're going to be taking a look at all of this. So try to keep that broad sort of perspective in mind as we're going through the details. First of all, let's talk about the stimuli, right? What types of stimuli can cells respond to? And as I said, they're very diverse. Even from your own experience, you know that you as a human being respond to all kinds of stimuli coming in all the time. You're able to have vision as a response as a consequence of your ability to detect changes in light. That's your visual system, right? Mechanical stress. How do you know that you are touching your pencil, or you can feel the sensation of a breeze against your skin or things like that? That's all down to the ability of specific cells in your body that can detect mechanical perturbation. So mechanical stress is a type of stimulus that cells can respond to. And, not only touch, but hearing is an example of that kind of stimulus response. Temperature, you can detect changes in temperature because you have receptors that change their shape and therefore their function as a consequence of changes in temperature. As we're going to see, there are some receptors that will change their behavior as a consequence of changes in charge in their local region. And so that would be a voltage stimulus that's being detected there. And then finally, the largest class of stimuli is probably chemical stimuli, right? So these are chemical compounds that can interact with cellular receptors, and by binding to those cellular receptors can cause some change in how they're functioning and what they're doing in the cell. And these chemical signals are often referred to as ligands. And so we'll use that term during our discussion, thinking about ligand interactions with receptors. And what types of chemicals are we talking about here? Well, we could be talking about nonpolar gases, things like nitric oxide, steroids, like the steroid hormones, estrogen, testosterone, progesterone. There are some fatty acids that can act as chemical signals and interact with receptors and cause changes in cell behavior. For example, the arachidonic acid pathway that's very important in inflammation and pain response. Many of the chemical ligands that stimulate receptors are derived from amino acids. This is something we touched on earlier in the course. Those neurotransmitters, things like adrenaline and dopamine, are derived from amino acid precursors. There are also chemical signals that are derived from nucleotide. We talked about ATP in the context of cellular energetics, but it turns out that there are receptors that respond to ATP. There are receptors that respond to compounds that are derived from ATP, things like cyclic AMP, which we'll see coming up in this discussion. And then finally, we have proteins that can act as chemical signals to stimulate a particular response in a cell. For example, insulin, the hormone which regulates glucose uptake by the cells in your body, is an example of a protein that is a chemical ligand. All right, so diverse cellular stimuli are going to be able to interact with cells and elicit a diverse range of cellular responses depending on what the stimulus is. That makes sense. One thing before we get started looking at some of the details of signal transduction pathways, one thing that's important to understand is that we're going to see some commonalities in terms of the intracellular pathways that we're going to be looking at. And one of the things that we'll see coming up over and over and over again is that when a cell surface receptor or even intracellular receptor is activated by some signaling molecule, a ligand, very often what's going to be activated within the cell to elicit the specific response is some sort of kinase or phosphatase or both cascade. Right? So you're going to see in response to a chemical ligand binding receptor, activation of some intracellular molecule that then is going to lead to activation of, let's say, a protein kinase. So we could imagine the first protein kinase in this cascade we'll call protein kinase 1. And that protein kinase in turn will phosphorylate and activate a stream of downstream proteins that will themselves have their own enzymatic activities associated with them, and often those downstream proteins are themselves kinases. So we might see something that looks like this, where protein kinase 1 gets activated, and it phosphorylates and activates a second protein kinase, which itself phosphorylates and activates yet a third protein kinase, and this can go on. And then somewhere at the end of that phosphorylation cascade, you're actually going to have some cellular protein, enzyme, structural protein, whatever, that gets phosphorylated by this last protein kinase in the chain, and this phosphorylation event here is actually what gives us our specific cellular response. Right now, as we've discussed before, this is a really broadly used, very commonly used mechanism for controlling enzyme activity and protein activity in a cell. This is a covalent linkage of a phosphate group to a molecule, which changes its structure and changes its function as a result. And in the context of enzymes, this is covalent modification leading to activation or inhibition, because it could go either way. Remember that as we discussed before, the source of that phosphate is going to be an ATP. So every one of these phosphorylation steps requires a transfer of a phosphate group from ATP to that target molecule. And we end up with ADP as a result. So in terms of energetics, this is a very energy costly process. Signal transduction is costly in terms of cellular energetics, right? The other thing that you need to remember is that, as we talked about before, for every kinase that exists in a cell and phosphorylates a target protein, there's going to be a corresponding phosphatase, just abbreviated here as PP, phosphatase enzyme, that's capable of cleaving that phosphate group off, right? Here's that phosphate leaving as an inorganic phosphate group, returning this activated protein kinase back to its inactive state. Right? And so there's an on switch in this case, the phosphorylation, and then an off switch, in this case, dephosphorylation by the phosphatase enzyme. And these are in competition with one another, right? The kinases are phosphorylating to activate in this example, and the phosphatases are dephosphorylating to inactivate. And it's the balance of activities of these kinases and phosphatases that determine whether this cellular response that's stimulated by this final player in the pathway is going to occur or not, right? So when this receptor is no longer being activated by its signaling molecule, then the kinases go back to their inactive state, and the predominant effect that we see here is dephosphorylation, because the phosphatases are removing those phosphates and there are no longer active kinases to add them back on. So these kinds of kinase and phosphatase cascades are really, really common in signal transduction, and we'll see several examples of this. Okay, so the next thing that I'd like to turn my attention to is discussion of the types of receptors that we are going to see coming into play here in our cell signaling pathways. And so firstly, let's just dispense with the more infrequent type of receptors that we'll see. And those are receptors that actually exist within the cell, what we might call intracellular receptors. They exist inside, they reside inside the cell, either in the cytoplasm or in the nucleus of a eukaryotic cell, right? And so these types of receptors are only possible when we're dealing with a chemical signal, a chemical ligand that is lipophilic. That is nonpolar and can diffuse directly through the phospholipid bilayer and enter the cytoplasm of the cell or the nucleus of the cell, if we're talking about that kind of pathway. Because these molecules can freely cross the cellular membrane, they can have intracellular receptors. And so one very common class of these intracellular receptors are those that respond to steroids. So steroid hormone receptors, the receptors that, for example, bind testosterone or estrogen, right? Those are steroid hormones. All steroid hormones are derived from cholesterol, and therefore they are nonpolar, largely lipophilic. They can slip right through the phospholipid bilayer. So what would happen is, let's say that this is an estrogen molecule. It would diffuse across the cellular membrane. It would have an estrogen receptor, we might call ER, estrogen receptor, to which it would bind. And this estrogen receptor, when estrogen binds to it, is capable of passing through a nuclear pore and getting into the nucleus of the cell. And typically, the traditional standard response that steroid hormones elicit is a change in gene expression. So the receptor, this estrogen receptor molecule itself, is itself a protein that binds to DNA and can cause an increase or decrease in gene expression. So these are referred to as transcription factors. Remember that the process of copying DNA to make mRNA is called transcription. And so these proteins that can affect the rate at which that copying occurs are called transcription factors. And so steroid hormone receptors are transcription factors. but they are transcription factors that can only affect gene expression when they have their steroid hormone bound to them. So how this would work is that the protein would bind to DNA, and in binding to DNA, we would see maybe an increase in the mRNA copied from that specific gene. That mRNA would then be exported out of the nucleus of the cell, would interact with ribosomes in the cytoplasm, and we would see synthesis of the proteins encoded by that gene. So the cellular response that is elicited by these types of signaling molecules, these steroid hormones, is a change in the types of proteins that a cell is making, right? And by changing the types of proteins that a cell is making, you're changing the functional capabilities of that cell. Since proteins are the workhorses of the cell that carry out the vast majority of cellular function, by changing which proteins are present, you're actually dramatically changing what that cell is capable of doing. And down here in the left, what you see is a structural representation of what that binding to DNA actually looks like. So here's our DNA double helix down here. You can make out those major grooves, the deep grooves in the backbone of the helix itself, and this ribbon up here would be the estrogen receptor or testosterone receptor, whichever steroid hormone receptor we're looking at here, actually sitting down and making close contact with the DNA and feeling out the sequence, which is sort of color-coded here by virtue of those very close interactions that the protein is able to have with those bases in the interior of the DNA helix, right? So transcription factors. Some receptor molecules are actually transcription factors that impact the rate of transcription of specific genes in the cell. The second type of receptor that I'd like to talk about are a set of receptors that exist in the membrane. So actually all of the remaining receptors that we're going to talk about are membrane-associated receptors. So these would be binding to or responding to ligands that do not cross the cellular membrane. And this next class that we're going to discuss are the gated ion channels. So as the name implies, an ion channel is a transport protein that allows ions to flow across the membrane, right? So if we imagine this is the plasma membrane of a cell, this is the cytoplasm, this is the extracellular space, you have some channel protein that exists in the membrane. But the difference here is that these are gated in such a way that they can exist in an open configuration or a closed configuration. So when the gate is closed, the specific ions that this membrane channel would ordinarily allow to pass are unable to pass through the protein, right? So gate closed, ions don't flow. These gates on the ion channel can be opened when the signaling molecule or ligand binds to that channel. So for example, here we have a ligand binding here, and the gate is opened up, and we see ions flowing across the membrane, and somehow that change in ion concentration on the inside of the cell is going to elicit some specific set of cellular responses. When that ligand is removed or the concentration decreases, then the gate is once more closed. And this is showing, a very dramatic sort of fence-like gate opening and closing across the pore that these ions move through, and typically the shape change that we see is a lot more subtle. So here's a more accurate representation of what a gated ion channel might look like. So in gray here, this would be the membrane of the cell. This particular ion channel is the acetylcholine receptor, and we're going to see in just a second how this works to allow control of muscle cell contraction by the nervous system. But what you can see is that it has multiple subunits. These are all color-coded here. And this is looking at it from the side, so it would be embedded in the membrane. And this is looking at it from the top down, so you can see this channel or pore that passes through the membrane. So this is where the ions would be flowing. And rather than having a gate that sort of opens and shuts like this, typically what happens with one of these gated ion channels is that this pore size is changed in response to ligand binding. And so it gets smaller or larger, sort of like the shutter on a camera opens and closes, that diaphragm effect opens and closes that channel. So acetylcholine receptor is the receptor that's found at the junction between neurons and muscle cells. So as you know, voluntary muscle contraction is regulated by the nervous system. Well, how does a nervous system cell, a neuron, regulate the contraction of a muscle cell, so a different tissue? Well, it does that by releasing a chemical that binds to the muscle cell and elicits that response. So let's see how that would work. Okay, so let's take a look at how this would work at the neuromuscular junction. How does a neuron control the activity of a muscle cell? So up here, this sort of squiggly looking cell is my neuron. And up here is the cell body where we would find the nucleus and all the major organelles. And what we would see if we were looking at it at a typical neuron is that it would have this very characteristic structure where coming off of that cell body, we would see a number of cellular processes sort of extending out in all directions. And these processes come in two basic types. So there are the dendrites, which we see up here. And those are connecting up to other cells, other neurons potentially, and receiving information and sort of transmitting that information towards the cell body. And so every neuron is going to have hundreds to thousands of these dendritic connections with other cells, constantly receiving information from those cells. And then typically what we would see is one long process coming off and making contact with one target cell, and this would be the axon. Now in this case, the type of neuron that we're looking at is a motor neuron. It's one that's going to be controlling voluntary muscle contraction, and so the target cell in this case is a muscle cell, right? And the way that a neuron gets stimulated by all of these dendritic connections is through voltage, basically. It's the flow of charges across the membrane, which generates an electrical signal referred to as an action potential, and that electrical signal is going to flow down this neuron, down the axon, to the region of the axon that is in close contact, not quite contact, but very close approximation to the target cell. And this is referred to as the synapse. So we'll have an electrical signal, an action potential flowing down this motor neuron. So this could be a stimulus, that's happening in your brain. You make a decision that you're going to move a particular muscle in a particular way. That's going to generate this action potential, this electrical signal down the motor neuron. And then what happens is that within this motor neuron, in the axon terminus, there are all of these vesicles. And the vesicles are filled with a neurotransmitter called acetylcholine. So here's the name written out, acetylcholine, and we're just going to abbreviate that as ACH. So within these vesicles, we have tons and tons of acetylcholine neurotransmitter. And when this action potential reaches this region of the neuron, these vesicles are induced to fuse with the membrane, right? And so we've talked about fusion before, and we know what happens when a vesicle fuses with the membrane. What was inside the vesicle is now released to the outside, right? And so now we have this acetylcholine neurotransmitter that has been released to the outside of the neuron into this synapse. Well, on the target cell, in this case on the muscle cell, there is a binding site for acetylcholine because these are acetylcholine receptors. And so as acetylcholine binds, that's going to trigger a change in the acetylcholine receptor, which, as we said, is a gated ion channel. And when the ligand binds, the gate opens, so that pore in the membrane gets a little bit bigger. And in this case, the ions that are going to be allowed to flow are primarily sodium ions. Now, why would there be sodium ions around to flow in any case? Well, remember that we talked before about the activity of the sodium-potassium pump? This sodium-potassium pump is going to be present in all the cells of your body. And remember, we talked before about this form of active transport. The energy of ATP hydrolysis is converted to transport of sodium ions and potassium ions across the membrane. More specifically, with every ATP that gets hydrolyzed, 3 sodium ions are pumped out, 2 potassium ions are pumped in. And so what this does is it creates a gradient where there is a lot more sodium outside the cell than inside, and a lot more potassium inside the cell than outside, right? So this is establishing a concentration gradient, which itself is a form of potential energy. So these sodium-potassium pumps, which are sort of color-coded here in red, are everywhere, and they're constantly building this gradient, right? Higher potassium in, higher sodium out. So when the acetylcholine receptor opens up, sodium ions are allowed to flow down their gradient back into the cell. And that sodium flux across the membrane introduces more positive charge into the interior of the cell. And that positive charge causes a change in the voltage around another ion channel, another gated ion channel. In this case, it is a voltage-gated calcium channel. So this is one of those channels that's gated by potential, electrical potential. So when the sodium ions flow in and we get lots more positive charge in the region of this voltage-gated calcium channel, then it opens up and forms a pore in the membrane, and everywhere where there's a gradient of calcium, we're going to see calcium being released. Well, if this is a muscle cell, where do we have high concentrations of calcium being stored? In the smooth endoplasmic reticulum. We talked about this in the fish lab, that the efflux of calcium out of the smooth ER is actually the stimulus for muscle contraction. So that's exactly what happens here. As sodium ions flow into the cell through the acetylcholine receptor, a buildup of positive charge around this voltage-gated calcium channel in the smooth ER causes the gate to open, and calcium is going to flow out of the smooth ER into the cytoplasm of the muscle cell, and as we talked about before, that triggers the interaction of our myosin motor proteins with our actin microfilaments in our cytoskeletal array that causes muscle contractions. And so these myosin motors connect up with our actin microfilaments and pull on them, and we see shortening of these sarcomeres, as they're referred to, and that's muscle contraction, right? So that's stimulated by calcium. The calcium is released from the smooth ER because of the positive charge that comes flowing in through the acetylcholine receptor when acetylcholine is released from the stimulating neuron and binds to that acetylcholine receptor. So just like we had the sodium-potassium pump that was burning energy, burning cellular energy, right, hydrolyzing ATP to establish the sodium gradient, similarly there would be an active transport protein, a calcium pump, that would be doing the same thing to build this gradient of calcium, storing calcium within the smooth ER and in other organelles in the cell as well, so that it could be released in a regulated, stimulated fashion and can trigger muscle contraction only under stimulation of the muscle cell. So in this way, a stimulated motor neuron that itself is under regulation by this electrical signal that has been generated within it releases a chemical signal to the exterior environment, which then binds to acetylcholine receptor on the target cell and triggers this internal set of responses. How do we stop muscle contraction? Because obviously it's going to be very important that we stop the muscle contraction, otherwise you get a condition called tetany where you have contraction with no relaxation, and that's bad. So there are a couple of things that go on here to terminate this signal. First of all, this acetylcholine molecule is degraded by an enzyme, which we can just represent by this little Pac-Man guy, that exists here in the synapse, and that enzyme is called acetylcholinesterase. So acetylcholinesterase. So what this acetylcholinesterase enzyme does is it degrades the acetylcholine in the cleft. It actually splits it into two molecules, acetic acid and choline, and in so doing removes the signal that's keeping these gated ion channels open, and so then they close. And then when they're closed, no more sodium is going to flow across the membrane, right? So step one in terminating this signal is degradation of the acetylcholine ligand by the acetylcholinesterase enzyme that's also present here in the synapse. The other thing that's going to happen to terminate this signal is that these sodium-potassium pumps are constantly working in the background to restore those sodium and potassium gradients, right? So pumping that sodium back out of the cell to restore our starting concentration gradients. Same thing with our calcium pump down here, burning the energy of ATP to take these calciums that were released and basically hoover them back up into the smooth ER and restore those gradients. So a lot of cellular energy is expended to build these concentration gradients to maintain the responsiveness of this motor, the responsiveness of this muscle cell to its controlling motor neuron. So in this example of cellular signaling, what we actually have are two different types of gated ion channel. The acetylcholine receptor is a ligand-gated ion channel, that is, its open or closed configuration is controlled by binding of the acetylcholine ligand, and a voltage-gated ion channel, this calcium channel, whose shape and behavior is changed by a change in charge in this local region around the protein. So two different types of gated ion channels, but there are lots of others that respond to different types of stimuli as well. So mechanical stimulus, temperature changes, things like that can also regulate these gated ion channels and allow the flux of charges across the membrane, which can cause other specific types of cellular response. And we'll see another example in just a little bit of calcium being used as a signaling molecule downstream of a different type of receptor. So not a gated ion channel, but a different type of receptor. All right, so moving on, we've talked about the gated ion channels. That's our first type of membrane-embedded receptor that we wanted to understand the function of. The second type of membrane-embedded receptor that we're just going to mention briefly is the receptor tyrosine kinase class of membrane receptor. And so receptor tyrosine kinases are, as their name implies, enzymes. They're kinases. And the way that they transmit a signal to the interior of the cell is via their kinase activity. They're called tyrosine kinases because they particularly target tyrosine amino acid residues within proteins. They're protein targets for phosphorylation. And all receptor tyrosine kinases function in the same basic manner, right? So they have an extracellular domain, which is where the ligand that's going to control the activity of this receptor would bind. They have a transmembrane domain that passes through the phospholipid bilayer, and then they have an intracellular kinase domain. The other thing that's in common with receptor tyrosine kinases, no matter which one you're looking at, is that typically one receptor being activated by ligand is not enough to generate a signal. In fact, you have to have two separate molecules of receptor tyrosine kinase both activated at the same time in order to transmit a signal to the inside world, right? So the way that this works is that in isolation and without ligand bound, these kinase catalytic domains are inactive. They do nothing. They have no enzymatic activity. When ligand binds to both of these receptors, so this little pink molecule here, we'll put an L on, that's our ligand. So it's bound to this receptor and it's bound to this receptor. It triggers association of these two receptors with one another. So they come together and they form a complex, and this is referred to as dimerization. They form a dimer, right? Di meaning two, mer meaning unit, so two units come together. And when they come together, the catalytic domains down here inside the cell are actually activated. And what happens is that this kinase domain on this receptor will actually phosphorylate this kinase domain and vice versa. So this is referred to as cross-phosphorylation. It's sometimes also referred to as autophosphorylation. And when these catalytic domains cross-phosphorylate one another, it changes the shape, it changes the structure of this internal kinase domain. So that's sort of represented here by this difference in structure. So you can see this little orange domain right here, which has a different structure in this representation. And these are representing phosphorylated regions of this protein. So we've added phosphate groups on, and that's changed the shape of this protein. These would be additional phosphate groups that have been added. Down here we've got additional phosphate groups. So lots of phosphorylation of these guys onto each other. And then what happens is that these phosphorylated domains within the cell can be bound by intracellular proteins that have particular affinities for these receptors, but only when the phosphates have been added. And so this would be some sort of a signal relay protein. And what it would do is, in binding to this phosphorylated domain, potentially it could be phosphorylated itself by the enzymatic activity of the receptor, and then it would translocate to the interior of the cell and affect other target proteins. So if you remember back at the beginning of the discussion when we were looking at that kinase phosphatase cascade, this would be an example of the type of signal relay molecule that could be impacted by a receptor tyrosine kinase and could trigger that kind of kinase phosphatase pathway within the cell. All right, but like I said, all receptor tyrosine kinases basically go through the same sort of dimerization and cross-phosphorylation process to generate these phosphorylated binding sites that now bind intracellular proteins and change their behavior and trigger some specific cellular response. And so that's all we're going to say about receptor tyrosine kinases. For the rest of our discussion of membrane receptors, we're going to focus on the class of membrane receptors that are called G-protein coupled receptors. G-protein coupled receptors, often called GPCRs for short. So we're going to see several examples of G-protein coupled receptors and the types of cellular responses that can be initiated by these receptors and the typical signal transduction pathways that are activated by these receptors. Just like we saw with the receptor tyrosine kinases, all G-protein coupled receptors function in the same basic way. They get activated in the same basic way, but as we're going to see, once they're activated, they can trigger a diverse set of intracellular responses within the cell and cause lots of diverse cellular responses as a result. So here is a simple cartoon diagram of what a G-protein coupled receptor would look like. They're very typically referred to as 7 transmembrane domain proteins, which, as you might imagine, is due to the fact that they have 7 transmembrane domains. So this is one receptor molecule, one polypeptide. Here would be the N-terminus of the polypeptide, and as you can see, it winds through the membrane once, twice, 3, 4, 5, 6, 7 times, and then this would be the C-terminus of that polypeptide. You can see your alpha helical transmembrane domains like we saw before passing through the bilayer. And so this outer region of the receptor, which is exposed to the extracellular environment, would be where the ligand that interacts with this specific receptor would bind. And then here on the intracellular face is the region of the receptor that's going to interact with an intracellular signaling molecule. And in the case of G-protein coupled receptors, that intracellular signaling molecule that's going to bind here, appropriately enough, is what's called a G protein. And we'll look at that in more detail in just a minute. Right? So all G-protein coupled receptors stimulate a cellular response initially by affecting the behavior of an intracellular G protein that interacts with the G protein coupled receptor. So that's why it's called a G protein coupled receptor. The receptor is coupled to the activation of a G protein inside the cell. So this is a more realistic representation of a real G protein coupled receptor. In this case, it's a beta adrenergic receptor. This is the receptor that is responding to something like, for example, adrenaline, epinephrine, and we're going to see how that works in just a second. So here would be the ligand binding domain up here. These would be the 7 transmembrane domains that pass through, and then here would be the intracellular domain that would interact with the G protein. So just to give you a better sense, it's not sort of arrayed in this linear fashion. Those 7 transmembrane domains are sort of smushed together in this fashion. All right, so how do G-protein coupled receptors work then? Well, they all work by the same basic mechanism. You have a G-protein coupled receptor in the membrane. It has and a set of G proteins that can interact with it, but only when the receptor has been activated by ligand binding, right? So the G protein is not embedded in the membrane, but it's what's referred to as a peripheral membrane protein. It's loosely associated with the intracellular face of the plasma membrane. So what happens is that a signaling molecule is going to bind to the G protein coupled receptor. So here's our GPCR, G protein coupled receptor, and that triggers a change in the behavior of the G protein that associates with that receptor. And in this case, the change in behavior is a change in the way this protein interacts with a molecule of GTP. So these G proteins are called G proteins because they can bind to GTP and hydrolyze GTPs. So GTP is a nucleotide just like ATP is a nucleotide, and as we've talked about before, GTP can be used in similar fashion to ATP in cellular energetics. So cleavage of a phosphate off of GTP is exergonic, just like cleavage of a phosphate off of ATP is exergonic. So you can see cleavage of GTP to GDP and phosphate, and that exergonic reaction can be linked to something else that's endergonic, right? In this case, it's linked to a change in the behavior of a G protein. So G proteins are G proteins because they bind to GTP and hydrolyze GTP. So in the unexcited state, in the unstimulated state, inactive G protein has a GDP bound, right? But when the GPCR has its ligand, its signaling molecule bound to it and becomes activated, it stimulates this associated G protein to release the GTP that it was holding. So it releases this, it lets go of it, and it binds a new molecule of GTP. And so now this G protein is holding a brand new GTP, right? And when this G protein is holding a GTP, what can happen is that it'll dissociate from the GPCR receptor and it'll be able to slide along the membrane and interact with downstream enzymes, specific enzymes that it is an allosteric regulator of. So activated G protein can allosterically activate or inhibit, in some cases, some downstream enzyme, which is then going to catalyze its specific response to lead to some kind of appropriate reaction in the cell. All right? Now, each of these G proteins, as we said, can not only bind to GTP, but it can also hydrolyze GTP. And that's exactly what happens, is that at some rate, this G protein is going to spontaneously hydrolyze the GTP to GDP and phosphate. The phosphate is released, but the G protein holds on to the GDP, and because it's no longer holding GTP, it goes back to its inactive state. Right? And so it can transition between this inactive state and this active state by virtue of the fact of whether it's holding GDP or GTP. So the G protein has a different shape in either of those two circumstances, but it's only going to release this GDP and pick up a new GTP when it binds to an activated receptor. All right, so the process is GPCR gets activated by a signaling molecule. It interacts with an intracellular G protein that's specific for that specific receptor. The G protein is stimulated to release the GDP it was holding and bind a new GTP. That leads to activation of the G protein, which will then allosterically activate or inhibit downstream enzymes in the cell to elicit some cellular response. Spontaneous hydrolysis of GTP to GDP and phosphate inactivates the G protein. It'll no longer be allosterically activating its downstream target enzyme, and we go back to the starting state, right? So termination of the signal in this case can be accomplished by spontaneous hydrolysis of the GTP molecule itself. So all G protein coupled receptors work via this same basic mechanism by activating a G protein within the cell. So what we're going to do next is we're going to look at three different very commonly used intracellular signal transduction pathways that are commonly linked to G protein coupled receptor activation. And the first one that we're going to look at is the adenylyl cyclase and cyclic AMP signal transduction pathway. So again, we're still in the world of G protein coupled receptors. The next three examples we're going to see are all G protein coupled receptors, but what we're seeing is those receptors linked to different intracellular signaling pathways, right? So in this case, what we're looking at is the adrenergic receptor, whose structure we just looked at. More specifically, it's the beta-2 adrenergic receptor. So this is the receptor that would respond to the signaling molecule adrenaline, or epinephrine, which, as I'm sure you've heard at some point, is the molecule, the hormone that's responsible for the fight or flight response in animals. So the basic idea here is that you encounter a situation, a scenario, which is potentially detrimental to your life. You have this hormone released from the adrenal glands, and it stimulates physiological responses in lots of different organ systems, all of which are designed to save your life, right? So maybe you've just seen a tiger stalking you through the jungle, and you have to outrun that tiger, and all of the physiological responses that adrenaline elicit in your body are designed to help you do that. So what sorts of responses? Well, increase in heart rate, increase in respiration, so that you can acquire more oxygen from the environment to power your cells via cellular respiration. But in order to have that cellular respiration, what are your cells going to need? They're going to need more glucose, right? So one of the specific responses that we see downstream of adrenaline stimulation or epinephrine stimulation is release of glucose in skeletal muscle cell, release of glucose from glycogen specifically, right? So this is referred to as mobilization of glucose. So you have stored glycogen within your muscle cell, and when that muscle cell receives a stimulus in the form of adrenaline signaling, the glycogen stores actually get cleaved, releasing lots of glucose into the cell, so the cell can use that for cellular respiration and outrun that tiger. All right, so the beta adrenergic receptor is a G protein coupled receptor. And so as we talked about before, the basic mechanism is going to be exactly what we described. The G protein coupled receptor is going to activate a G protein, which is then going to activate some downstream enzyme. In the case of adrenaline, the G protein, once it's activated, exchanges GDP for GTP and interacts with the downstream enzyme adenylyl cyclase. And it turns out that what this enzyme does is it takes a molecule of ATP and converts it to a molecule of cyclic AMP. And this molecule of cyclic AMP is referred to as a second messenger. So if we consider the original hormone signal that bound to the receptor as the first messenger that stimulated the initial response, this would be a second messenger. And in general, what we were referring to when we say second messenger is some small, diffusible, non-protein molecule that is either released or synthesized as part of a signal transduction pathway, and that will then relay the signal from the cell surface to the interior of the cell to elicit some specific response, right? So that's what we mean by second messenger. Cyclic AMP is one of the most commonly used second messengers in cells. So let's see what happens when adenylyl cyclase is activated downstream of the adrenaline G protein coupled receptor. Synthesis of cyclic AMP is actually going to activate this first protein kinase called protein kinase A. And in different signaling pathways, protein kinase A can interact with a lot of different intracellular proteins and affect their activity. In the context of releasing glucose from glycogen stores in skeletal muscle, what happens is that downstream of protein kinase A, what we see happening is that kinase phosphorylates and activates a second kinase. This one is called phosphorylase kinase. Phosphorylase kinase then phosphorylates and activates a third enzyme, which is glycogen phosphorylase. Glycogen phosphorylase is the enzyme that's actually responsible for cleaving glycogen to release glucose in the form of glucose 1-phosphate. And then there's an enzyme in the cell that will isomerize glucose 1-phosphate to glucose 6-phosphate, which, as you'll recall, is the starting substrate for glycolysis. Remember that the first step of glycolysis is phosphorylation of glucose to glucose 6-phosphate? Well, here it is, glucose 6-phosphate. Once we have it, we can feed that right into glycolysis, and in a skeletal muscle cell, what is that going to lead to? Lots and lots of cellular ATP, so that, again, you can outrun that tiger. All right. Now, how does cyclic AMP activate protein kinase A? Well, this itself is actually an interesting bit of regulation that takes us back to our discussion of allosteric regulation, because it turns out that cyclic AMP is an allosteric regulator of protein kinase A. So protein kinase A is a protein complex, actually. It contains 4 subunits. So 2 subunits that we're going to sort of represent by these little circles and a C. These are the catalytic subunits of protein kinase A. And then 2 subunits that we're going to represent by these little boxes, and these are the regulatory subunits. Right? So we have 2 catalytic subunits, 2 regulatory subunits, and the regulatory subunits actually inhibit the kinase activity of the catalytic subunits. So as long as the regulatory subunits are bound to the catalytic subunits, then protein kinase A is inactive. So the way that cyclic AMP activates protein kinase A is by binding to the regulatory subunits. So here's our R, binding to the regulatory subunits and causing them to release from the catalytic subunits. So when the catalytic subunits are released, now they are active. So this is active protein kinase A. Right? So as long as cyclic AMP is around, cyclic AMP is going to bind to these regulatory subunits and cause the release of the catalytic subunits, which are what the activated protein kinase A actually consists of. All right, so let's take a look at in a little bit more detail about cyclic AMP and how it's synthesized and how it's degraded. So here's our molecule of ATP. Here's our adenosine. Here's the adenine nitrogenous base. Here's the ribose sugar and our 3 phosphates. So here's ATP. The activity of adenylyl cyclase is to cleave off these 2 terminal phosphates. So these 2 terminal phosphates are released as a pyrophosphate group, and the remaining phosphate actually gets cyclized, so we get a second bond here with the number 3 carbon of the sugar. So it's referred to as cyclic AMP because it's been cyclized, or we've had a new ring structure introduced here where the phosphate is attached at 2 places to our sugar. So this is cyclic AMP. So this is what would then activate our protein kinase A by inhibiting the regulatory subunits, which are themselves inhibiting protein kinase A activity. So just as we talked about for our kinase and phosphatase pathways, where we had kinases that were activating something and phosphatases that were inactivating, here we have adenylyl cyclase synthesizing cyclic AMP. There's an enzyme that actually inactivates cyclic AMP and converts it to plain old vanilla AMP. And that enzyme is phosphodiesterase, which hydrolyzes this bond, this second bond that was introduced. And when you hydrolyze that bond, what you're left with is adenosine monophosphate. And so this adenosine monophosphate has no effect on protein kinase A's regulatory subunits. It's released from the regulatory subunits, and protein kinase A is once again bound by those regulatory subunits and inactivated. So in terms of our termination of our signal, in this case, we actually have several levels of termination that go on here. So phosphodiesterase is going to get rid of our cyclic AMP, which is going to in turn inactivate our protein kinase A and send it back to the inactive state, right? As we talked about before, at a spontaneous rate, this G protein is going to hydrolyze the GTP that it's holding, and that is going to inactivate the G protein, which will then no longer allosterically activate the adenylyl cyclase. So that shuts off the signal here. At the end of our discussion, we're actually going to talk a little bit more about termination of this signal at the level of the receptor, because it turns out that there's actually a third way that this signal is terminated, and that involves taking this whole receptor ligand complex into the cell, which is referred to as receptor internalization. And we'll talk about that more in a little bit. Internalization. By removing that receptor and the ligand from the cell surface, we're also switching off the signal. All right, so we'll return to that at the end of our discussion and see how that works in more detail. Okay, so that's cyclic AMP, protein kinase A pathway. Very, very commonly used in lots of different contexts, not just the context of response to adrenaline. Now, this seems needlessly complex, right? If the whole intent here is to have adrenaline bind to this receptor and activate glycogen phosphorylase to cleave glycogen, why not just have glycogen phosphorylase bind here to the receptor directly and get activated, right? Why go through all of this rigmarole of all of these other intracellular signaling molecules that have to be activated upstream of glycogen phosphorylase? And if you think about it a little bit, you can see that there would be definite advantages to doing it this way, the more complicated way, than a direct activation. And one thing is that we could actually have branching of this pathway. So as I said, protein kinase A doesn't only interact with this set of enzymes that leads to glycogen release. It's also going to be able to interact with other intracellular signaling molecules, right? So we can have a divergence of our pathway where one of our activated intermediates here can cause a number of different things to happen. So that's one thing. Likewise, by having more intermediates along the pathway, we potentially can have interactions or inputs from other signaling pathways in the cell. So we increase the amount of crosstalk, essentially, that goes on between intracellular signaling pathways by having multiple steps. But the other thing that happens when we have a multi-step pathway is that we have the capacity to amplify the signal, right? So if you imagine a scenario where you have your epinephrine binding to your adrenergic receptor and activating one molecule, so what we could have here is our glycogen phosphorylase binding directly to this adrenergic receptor, and maybe we'd see a couple of hundred glycogen phosphorylase enzymes activated if we did it that way, through that direct interaction approach. But instead, what actually happens is that this receptor is going to interact with maybe 100 G proteins, and each of those G proteins can bind to and activate an adenylyl cyclase. But each of those activated adenylyl cyclases can synthesize many cyclic AMP molecules. So we get amplification of the signal here, where we go from 10 to the second to 10 to the fourth cyclic AMPs, which each can activate protein kinase A, right? So we get 10 to the fourth activated protein kinase A's. Each of these activated protein kinase A's can activate many downstream phosphorylase kinase enzymes, each of which can activate many downstream glycogen phosphorylase enzymes. And so what we see is at each step here, amplification of the signal by virtue of the fact that each of these enzymes can target multiple substrates, many substrates, tens or hundreds of substrates, for catalysis, right? And so at the end of this, we end up with a ton of glucose 1-phosphate released to be able to feed into glycolysis, and that's a huge benefit to the cell. So in terms of efficiency and amplification of the response, having a multi-step pathway with amplification all along the way is much more beneficial to the cell in this type of a context, right? Okay, so the second G protein coupled receptor pathway we're going to look at also relies on calcium to transmit or transduce the signal and stimulate the cellular response in the cell, right? But it works a little bit differently. So we saw calcium stimulating muscle contraction before. Here we're going to see calcium activating enzymes and leading to a specific cellular response in that context. So once again, just to remind you, the only way that this works is by virtue of the fact that the cell expends a lot of energy to pump calcium out of the cytoplasm and lower calcium concentrations in the cytoplasm relative to other cellular compartments. So we talked before about smooth ER being a reservoir of calcium. It turns out that mitochondria are also have active transport of calcium into the matrix, and so that's another reservoir of calcium in the cell. And then also transport of calcium out of the cell into the extracellular space happens as well. So take-home message here, calcium concentrations in the cytoplasm are low, and we have these other sort of reservoirs or high concentration areas of calcium in the cell, which under stimulation can see calcium being released into the cytoplasm and causing some specific response to occur. So downstream of acetylcholine receptor, we saw that that was muscle contraction. Here we're going to see calcium being used as a second messenger downstream of a G protein coupled receptor. Now in this case, the specific G protein coupled receptor that we're going to be looking at is the glucagon receptor. Glucagon is a hormone that's released by the pancreas when levels of blood glucose are low, right? So let's say it's been a long time since you've had a meal and your sugar levels, your blood sugar levels are low, and your body cells are starting to really crave glucose, right? They've used up their intracellular stores of glucose and they need more glucose to keep cellular respiration going. Well, where are they going to get that glucose? As we've talked about before, you have two main storage depots for glycogen in your body. The first is muscle, and those muscle cells are really storing glycogen for themselves, right, to use in an emergency when glucose runs out. They're very high energy need tissue, and they basically have their own storage depots of glycogen. But for the rest of your body cells, the liver is responsible for storing that glycogen. And so when blood sugar levels are low, the pancreas releases this glucagon hormone, which binds to a glucagon receptor, which is a G protein coupled receptor on liver cells, and stimulates a response in the liver cell that ultimately leads to release of glucose by the liver. Right? So the glucose is released to the bloodstream. The rest of your body cells now have access to that glucose and can use it for cellular respiration. So this pathway, as I said, ultimately is going to release calcium, but it actually uses two different second messengers. Calcium is one, and the second is a molecule called IP3. So there are two separate second messengers in this case, two small diffusible non-protein molecules that transduce the signal. So as we talked about before, the basic mechanism of activation is going to be the same. We have a G protein coupled receptor that gets activated. That leads to its G protein exchanging GDP for GTP. Whereas we saw before adenylyl cyclase being activated, in this pathway what gets activated is an enzyme called phospholipase C, or PLC, phospholipase C. And if you think about the name of this enzyme, phospholipase, it's an enzyme that actually cleaves a phosphorylated lipid-associated molecule. And that lipid-associated molecule is something called PIP2, for phosphatidyl inositol bisphosphate. You don't have to remember that. All you have to remember is that this is basically a phosphorylated sugar. This is an inositol sugar, and these are phosphate groups. And this phosphorylated sugar is attached to the membrane by a little membrane-embedded molecule called diacylglycerol, or DAG. And what phospholipase C does is it cleaves, hydrolyzes the bond between this phosphorylated sugar and that little membrane-embedded diacylglycerol, or DAG, and releases this from the membrane. So now it's free to diffuse, right? This is a polar molecule. It's a sugar with 3 phosphate groups on it. And now that it's cleaved away from the membrane, it's called inositol triphosphate. Inositol triphosphate. And This inositol triphosphate is a second messenger, and it's now free to diffuse within the cytoplasm of the cell and interact with target molecules. In the context of this particular pathway downstream of glucagon, the target molecule that it interacts with is a gated calcium channel on smooth endoplasmic reticulum. Now, in the case of muscle contraction, we saw a voltage-gated ion channel, a voltage-gated calcium channel. In this case, we see a ligand-gated calcium channel, and the ligand is IP3. When IP3 binds to this calcium channel in smooth ER membrane, the channel opens up, calcium falls down its concentration gradient into the cell cytoplasm, and now calcium can act likewise as a second messenger, right? So IP3 was our first second messenger, which triggered calcium release from smooth ER. Now calcium itself can act as another second messenger, diffusing through the cytoplasm of the cell and interacting with lots of different target molecules. In this case, it interacts with a protein called calmodulin. Calmodulin is a protein that regulates the activity of a lot of cellular enzymes, and it does so in a calcium-dependent fashion. So in other words, calmodulin can allosterically interact with target proteins, but only when calcium is bound to it. So when calcium is released from smooth ER, calcium binds to calmodulin, and calmodulin can then go and allosterically activate a whole host of cellular enzymes and other proteins in the cell. In the context of this response, the cellular enzyme that ultimately gets activated is that same glycogen phosphorylase enzyme that we talked about in skeletal muscle cell. So glycogen phosphorylase, when calmodulin binds, cleaves glycogen to yield glucose 1-phosphate. That glucose 1-phosphate is isomerized to glucose 6-phosphate. Now remember, we're not in a muscle cell, we're in a liver cell, and the intent of hydrolyzing this glucose off of glycogen is not to feed the liver cell, but to feed the rest of the body. And so this glucose needs to be transported out of the cell. So the next thing that actually happens is that there's a phosphatase that cleaves that phosphate off, and we end up with plain old glucose, which can be transported by membrane transport proteins out of the liver cell into the bloodstream, where the rest of the body cells now have access to that glucose as a fuel source for cellular respiration. Okay, so we've seen now two specific examples of G protein coupled pathways that are commonly used in signal transduction. We saw the cyclic AMP pathway, the adenylyl cyclase cyclic AMP pathway, where cyclic AMP was used as a second messenger. And we just saw an IP3 coupled pathway where both IP3 and calcium are used as second messengers. In the next example, we're going to see yet another intracellular response to activation of a G protein coupled receptor. And that's going to involve activation of an enzyme called a MAP kinase. Okay? And the context that we're working in here, or that we're looking at here, is yeast mating. So this is something that you might not have even been aware that exists, that unicellular organisms can mate with one another, but yeast are special. So yeast are special in a number of ways, but one way that they're special is that they can either divide by a clonal expansion of the cell, so essentially one cell clones itself to produce multiple copies. And in that case, what we would be dealing with would be a diploid cell containing 2 copies of its genetic information, cloning itself, making an exact duplicate daughter cell, right? Set of daughter cells. But in some circumstances, those yeast cells can actually be induced to sporulate, to undergo meiosis and produce what are essentially gametes, cells that have only 1 copy of their genetic information. And when they do that, those cells that are gametes can actually fuse with or mate with other gametes, other yeast gametes, to produce another diploid cell. And so essentially what you see are haploid, so these would be haploid, only 1 copy of genetic information, what we might think of as gametes, reproductive cells, right? Even though these are yeast, so it's a little bit weird to think of them as gametes, but that's what they are. So they're spores, essentially. And so in finding a complementary partner, these gametes, these haploid cells that have only one set of genetic information, can find each other, mate with one another, fuse with one another, and form a cell that is diploid. 2 copies. Right? So haploid cells have 1 copy of genetic information, diploid cells have 2 copies of genetic information, and as long as nutrients are plentiful, then these diploid cells will just clone themselves and clone themselves and clone themselves and reproduce by producing identical copies of themselves, no problem at all. And they do this by budding. They just bud off a identical little cellular copy. When nutrients become limiting, so that's sort of indicated here by -n, minus nutrients, so under starvation conditions, this diploid organism will undergo a cell division that will result in 4 haploid cells, 4 spores or gametes, which can sort of go back and begin the process again, right? Find a mating partner. And if you think about it from an evolutionary perspective, it makes sense that this would be something that would be triggered by nutrient limitation. So environmental stress is being introduced here, nutrients are limiting. Maybe it's a good time to find some diversity in the population, some genetic diversity, and mating and combining genetic information from more than one cell is a really good way to mix up the genome and generate some genetic diversity. So that's the sort of broad overview of the perspective that you need to understand. But what we're going to do now is look at the details of how this happens. So it wouldn't make any sense for these cells to mate with a gamete or another haploid cell that is identical, genetically identical. And so one way that yeast cells can tell if they're mating with an identical cell or a different cell is by virtue of the types of mating pheromones that are produced. And so there are 2 types of mating pheromones that are possible, and those are the A-type pheromones and the alpha-type pheromones. And each of these haploid gametes will produce one, but not the other. And so that's sort of illustrated here by the 2 color codes, right? So this cell can produce either the A mating factor or the alpha mating factor. This cell can produce either the A or the alpha. And mating will only happen if the 2 cells involved produce different mating factors. So that's sort of an indicator of genetic diversity, right? That they produce different mating factors. So if this is a cell that produces the A mating factor, it will only respond to the alpha mating factor, right? So it will only produce receptors that respond to the opposite mating factor. And likewise, if this cell produces alpha mating factor, then it will only make receptors to respond to A mating factor. So cells produce a pheromone, and they only produce receptors for the opposite mating pheromone, right? Not their own. And that receptor is what's going to allow them to detect the presence of a cell in their environment that has the opposite mating type. And so as you can see here, these 2 cells are producing the pink, right? So maybe that's alpha, and the green, maybe that's A, pheromones. This one, let's say, produces the pink pheromone, and this one produces the green pheromone, and each one has cellular receptors that are only capable of responding to the opposite mating factor or pheromone type. So what happens when a yeast cell like this detects the presence of the opposite mating pheromone type? Well, in the region where that mating pheromone concentration is higher, that is, in the region where the cell is closer to that opposite mating factor cell type, what we see is the cell actually growing in that direction. Right? So each of these cells, by being stimulated by a cell producing the opposite mating type, actually extends out its cell wall, extends out its plasma membrane. The cell gets bigger, it grows in the direction of that mating partner. So this cell is growing in that direction, this cell is growing in that direction, and this polarized growth is referred to as schmooing. And the name of that comes from a cartoon character back in the 1940s called the Shmoo, which had this sort of pear-like elongated structure. And as you can see, if you look in the light microscope at yeast cells that are undergoing mating, they form these sort of Shmoo-like structures as they elongate in the direction of their mating partner. Eventually, they get close enough to their mating partner where the cell wall is going to come into contact. And what that induces is an enzymatic degradation of the cell walls to make kind of a pore in the cell wall so that the plasma membrane can now fuse, and what you end up with is one elongated cell, and you have the two nuclei of those cells that fuse and form one diploid cell with two copies of genetic information. All right, so that's the process of yeast mating and shmooing in general, in sort of broad overview. But what we want to do now is sort of zoom in to the microscopic level and think about what's happening here at this receptor level to trigger this sort of growth and reorganization inside the cell so that these cells can come into contact with one another. And this involves a G protein coupled receptor. So it turns out that the A and the alpha mating factors both bind to G protein coupled receptors of their own specific type. So this mating factor pathway is a G protein coupled receptor pathway. Like all G protein coupled receptor pathways, remember what's going to happen is that when the receptor gets activated, that's going to stimulate this G protein to exchange GDP for GTP. And then this activated G protein is going to stimulate the activity of some downstream signal transduction pathway, right? So we saw examples of enzymes that were activated, first adenylyl cyclase and then phospholipase C. The transduction cascade that's initiated in this example is a lot more complicated. And so we're actually going to leave out a bunch of the intermediate steps and just focus on an enzyme that gets activated downstream from that, and that's the enzyme Fuse3. Fuse3 is a kinase, and it's referred to as a MAP kinase. So it's sometimes abbreviated as a MAPK. All right? It is a kinase that phosphorylates target proteins. MAP stands for mitogen activated protein. So a MAP kinase is a mitogen activated protein kinase. And there are lots and lots of MAP kinases that are found in a lot of different types of eukaryotic organisms. A mitogen is anything that stimulates cell growth or cell division. So in general, these mitogen activated protein kinases, these MAP kinases, are functioning downstream of some signal that is activating growth or cell division, right? In this case, it's activating growth of the cell in a directional manner. So this MAP kinase, Fuse3, is phosphorylated, right? So now we have phosphorylated Fuse3, and it's now activated. So it's now an active protein kinase. So this phosphorylation step activates Fuse3, and Fuse3 is going to hit a lot of different target proteins. But one in particular that we're interested in is this protein called formin. And it turns out that what formin does is that when it gets phosphorylated by Fuse3, it can interact with the ends of microfilaments, and it can stimulate the addition of new actin subunits onto the microfilaments, and therefore cause the microfilaments to be extended, right, and reorganized. So this is essentially reorganization of the cytoskeleton, and where we see this happening is in the region of the cell where the mating partner is located. So this is actually what leads to this extension of the cell in that direction. So what we would have inside the cell is delivery of vesicles along this track, addition of new cell membrane and new cell wall components here, and so growth in that direction towards the mating partner. That would be up here. Now, I've given you in the Blackboard folder, the lecture materials folder, another video to watch that describes some experimental evidence for vesicle delivery as being an important part of this schmooing response and the ability of a yeast cell to grow in a directional manner, in a polarized manner, in a particular direction, which is going to be in response to detecting a gradient. So make sure that you watch that video because it's going to fill in a few more of the details regarding how the cell actually grows in that direction towards the mating partner and some of the experimental evidence that leads us to understand how that occurs. All right, so again, in this case, a G protein coupled receptor is actually triggering an intracellular kinase cascade. And just, a little interesting note here, this is a MAP kinase. And like I said, MAP kinases are found in a lot of different contexts for cellular signaling contexts. But these steps, these upstream steps that we're leaving out, very often involve kinases that are phosphorylating these MAP kinases. And so we have sort of a ridiculous situation where we have this MAPK that itself gets phosphorylated. And what do you call an enzyme that phosphorylates a MAP kinase? Well, you call it a MAP kinase. So that would be a MAPKK. These MAP kinases are themselves phosphorylated by upstream kinases. And what do you call a kinase that phosphorylates a MAP kinase? Well, of course, you would call it a MAP kinase. And in kind of a ridiculous fashion, this can actually extend up a significant way where you can have multiple MAP kinases in a row, ultimately, finally terminating on some important kinase that actually leads to some particular cellular response. So this is in the context of yeast mating, and the cellular response that we get is the interaction of this formin protein with microfilaments in the cell in such a fashion to stimulate the addition of new actin subunits and extend the cytoskeleton in this region where the cell is actually growing towards a mating partner. All right, so that's given us 3 specific intracellular signal transduction pathways that are downstream of G protein coupled receptors. These are some of the most important cellular receptors that exist, very, very often targets of pharmaceutical intervention when you want to change the behavior of cells and tissues and organ systems. A lot of times what's being targeted are G protein coupled receptors or the intracellular pathways that are downstream of those receptors. And as you can see, they, we share them all the way back to yeast, and they're evolutionarily very ancient signaling receptors. So what I want to talk about next is actually the final signal transduction pathway that any cell in a multicellular organism would go through, and that's apoptosis. So apoptosis is a signal transduction pathway that leads to cell death. And it's referred to as programmed cell death, or sometimes referred to as cell suicide. And it's essentially a mechanism that induces a cell to destroy itself, right? And there are a lot of contexts in which this is important. So it could be initiated in response to the cell detecting that its own behavior internally is abnormal. And so within the cell, there are mechanisms, molecular mechanisms to detect whether the DNA integrity is good or not, right? So monitoring the integrity of the DNA and things like chromosome shortening or DNA damage that can't be repaired can actually send a signal within the same cell that there's been DNA damage that can't be fixed, and this cell should remove itself from the population before it incurs a mutation that could potentially be harmful to the organism as a whole. So it's sort of an altruistic mechanism. A cell kills itself before it can cause problems for the organism as a whole. Other types of intracellular signals that can induce this response would be something like extensive protein misfolding. So in the rough ER, if there's a lot of misfolding going on, then that could indicate that there's something fundamentally wrong with the protein processing machinery. And then abnormal cell division. So cells can actually detect when their intracellular signal transduction pathways that regulate cell division are acting in an aberrant fashion. In other words, stimulating too much growth and too much cell division. And when that happens, that can also be a signal for the cell to undergo apoptosis or programmed cell death. In addition to sensing internally within the cell, this can also be induced from outside the cell. So you can have a death ligand that's released from one cell. Typically this would be something like an immune cell, but not always. And that immune cell can actually stimulate a target cell to undergo programmed cell death. Now, the nice thing about a cell dying this way, as opposed to the type of death that you see in response to injury or infection or things like that, is that this is what's referred to as a clean death. Right? So here's a normal cell, and if this cell actually was killed as a result of mechanical injury or infection or something like that, then it would undergo a messy death, where the cell would actually break open and lyse and release internal contents to the exterior environment. And if you think about it, what's inside a cell? Well, there are a whole bunch of enzymes in the cell. There are proteases. The lysosome contains, tons of hydrolase enzymes that would be released. So any cells that were outside in the neighborhood here would be coming into contact with all of those intracellular enzymes and would be negatively impacted by that, right? So that'd be a dirty death. With programmed cell death, what happens instead is that this cell essentially sort of implodes. So here's a cell that's undergone apoptosis. And essentially what happens is that this cell undergoes fragmentation. The nucleus breaks down and fragments, but the cell doesn't break open. So the cell fragments and forms these little blebs or vesicles that contain all of the cellular contents and prevent them from leaking out of the cell and exposing neighboring cells or surrounding cells to potentially harmful enzymes or chemicals. Once the cell has undergone this fragmentation, the cell surface is no longer the same as it was here. And the sorts of cellular membrane components and things like that that are exposed on the exterior actually attract phagocytes to migrate to the area where this apoptotic cell is and essentially engulf it, endocytose it, and eat it and remove it in a very clean fashion. So it would, essentially be doing cleanup duty to get rid of this apoptotic cell. So this is referred to as a clean death, and this is mediated by intracellular proteins called caspases. So caspases are a specific class of proteases, which, as we've learned before, are enzymes that hydrolyze proteins. Now, these proteins that get hydrolyzed specifically are the proteins that are making up the cytoskeleton, the proteins that are making up the nuclear lamina. So the proteins that are giving this cell, up here, this healthy cell, its nice spherical cellular architecture, when those proteins get hydrolyzed and degraded, then you can see that the architecture of the cell breaks down and we see this fragmentation and blebbing start to occur. So it's a very complicated signal transduction pathway, but these caspases play a very critical role in degrading the interior cellular structure and allowing that blebbing to take place. So when would we see apoptosis happening? Would it only be if there's a problem? Well, it turns out no, that you can actually have and do have apoptosis happening during normal development and normal physiology as well. So during development, during animal development, for example, you see apoptosis playing a role in a lot of different contexts. So tail regression, why don't we have tails? Well, even though in earlier embryogenesis we did have a sort of primitive tail structure, the regression of that tail during development is stimulated by apoptosis. So those cells are stimulated to undergo programmed cell death and essentially remove themselves from the embryo. How do we go from what's referred to as a limb bud, where our hand or foot is going to be early in development, to a mature limb that actually has distinct digits that allow us, that capacity to manipulate things. Well, the tissue that's in between the digits is stimulated by extracellular signals to undergo apoptosis, and when that tissue dies, then what we're left with are these individual digit structures, right? Sometimes that process fails. You get a failure of apoptosis, and somebody can be born with webbed fingers or webbed toes, right? It's not an infrequent event, but normally during development, those intervening structures are stimulated to undergo programmed cell death and remove themselves from the embryo. Another place where apoptosis is really important in development is during development of the central nervous system. So it turns out that during fetal development, we actually get way more neurons developed than we actually need, and way more connections or synapses between our neurons formed than we actually need, or that allows good, efficient communication to take place. And so towards late fetal development and in the early postnatal stages, the synapses that have been set up actually get pruned back, and surplus neurons that were created are eliminated through apoptosis. And so the number of neurons that we had initially actually shrinks a little bit. And this is really important for normal brain development. And if there's a defect in this apoptotic pathway, then you end up with too many neurons, too many synapses, too much communication that's actually leading to inefficient communication, and that is associated with cognitive disability. Right? So it's not only found in disease, apoptosis is a normal part of development as well. And it turns out that the ability of the immune system to trigger apoptosis is something that happens all the time, and it's something that's maybe a little alarming to think about, but we develop cancer every single day in our body. We develop cells that are going rogue and undergoing inappropriate cell division, and those cells are typically targeted by our immune system and stimulated to undergo apoptosis. And a cancer cell, or a potentially cancerous cell, that has a defect in the apoptotic pathway that makes it so that it can't be stimulated to undergo programmed cell death, is a cancer cell that could potentially become dangerous and harmful. And so disruption of the apoptotic pathway is an area of very active research in the cancer research community because of that fact, right? So cancer often has ways of not being triggered to undergo apoptosis. But under normal circumstances, apoptosis is what keeps our cells behaving like good citizens in the context of a multicellular organism that requires, all of our cells to be regulated and integrated in their function. All right, so zoom back out and let's think about the sorts of responses that we have been able to take a look at so far. So we've seen both nuclear and cytoplasmic responses that get triggered by cellular signaling. So it could be synthesis of enzymes or other proteins by regulating gene expression. So we saw that in the case of our steroid hormones. That would be a slow response, right? It takes time for that upregulation of transcription to occur. It takes time for those mRNAs to be then translated to build protein. So that's a slow response. We also saw fast responses that could be initiated through the activation and deactivation of enzymes. If you are targeting an enzyme for phosphorylation, that's pretty quick, right? That's flipping an on switch on an enzyme that's already present in the cell. We saw that signaling can lead to vesicle fusion, so secretion and membrane protein localization in the context of cellular signaling. We saw that rearrangement of the cytoskeleton and reorganization of the cytoskeleton leading to a change in cell shape. Reorganization or rearrangement in the context of muscle cell contraction by virtue of the fact that myosin proteins are pulling on microfilaments, and rearrangement of the cytoskeleton in the context of yeast schmooing, where the actin microfilaments were being extended out and reorganized in the region of the cell that was growing towards the mating partner. And then finally we saw that sort of last signal transduction pathway that would ever be initiated in a cell, and that is the apoptotic program or programmed cell death or cell suicide. And so that, that's sort of a broad representation of the different types of cytoplasmic and nuclear responses that can be elicited. One thing that we need to keep in mind is that every cell, is receiving multiple signals all the time. It's not just receiving one signal and figuring out an appropriate response, but multiple signals coming from lots of different directions that are giving the cell important information, and the cell has to integrate those signals. So we can just represent that sort of integration very, very simply by thinking about a cell that we have here that requires signals A, B, and C. If signals A, B, and C are received by that cell, then that represents a survival signal. And essentially the cell just keeps on keeping on, right? Doing exactly what it was doing before. If signals D and E are added in on top of signals A, B, and C, then maybe that's an additional signal that stimulates the cell to not only survive, but divide and produce new cells of the same type. Maybe in combination with two different signals, F and G, instead of just dividing, now this cell is being stimulated to differentiate, to specialize, to change its structure and properties in some way, to acquire some new specialized function. And then, of course, if these are survival signals, loss of those survival signals might actually be a signal for that cell to undergo apoptosis and remove itself from the population. So integration of signaling pathways is something that's happening all the time and is really important for cells to be able to get complex information about their complex environments. All right, so let's return to this topic of termination of the signal that we've sort of been referring to throughout our discussion and bringing up in each of our specific pathways that we looked at, how termination is taking place. But just to sort of recap what we've seen, so if we start up here at the top, termination can take place at the level of the receptor, so the ligand receptor interaction. When the ligand is no longer associating with the receptor, then the receptor goes back to being inactive, right? So there are a number of ways that that can happen. We saw that in the context of the acetylcholine receptor, there's actually an enzyme, acetylcholinesterase, that degrades the acetylcholine ligand. And by degrading it, removes the ability of that ligand to interact with the receptor. It's no longer around. We're going to see in just a minute more about receptor internalization that I mentioned previously in our discussion of G protein coupled receptors. But I just want to mention briefly that in some cases where you have one cell stimulating a nearby target cell with a ligand, sometimes the way that that signal can be shut off is by the ligand being taken back up into the cell that originally released it. And that's referred to as reuptake. Very, very common in the context of the nervous system in neuronal signaling, where neurotransmitters are released by a cell and have the ability to interact with the target cell, but then the releasing cell actually transports the ligand back inside. And so that's another mechanism that we didn't really look at a specific example, but I just wanted to mention that it does exist. All right, we'll come back to receptor internalization, but I just want to remind you of the intracellular off switches that we saw at play. So we talked about the fact that G proteins will spontaneously hydrolyze GTP to GDP, and the rate at which that occurs is the rate at which the G protein is inactivating itself. So it's sort of a self-limiting activation for those guys. We also talked about the fact that whenever there's a kinase pathway, there is a corresponding phosphatase that goes along with that, and those phosphatases are capable of removing the activating phosphates. So functioning essentially as the off switches, right? In the context of the cyclic AMP signal transduction pathways, we saw adenylyl cyclase synthesizes cyclic AMP. We also saw that phosphodiesterase cleaves cyclic AMP to adenosine monophosphate and thereby inactivates that pathway. With IP3, we didn't really mention this during our discussion, but it turns out that the way that this is inactivated is through hyperphosphorylation. So that inositol triphosphate sugar gets additional phosphates added on, and these molecules that have 4 or 5 phosphates don't interact with the ligand-gated calcium channel, and in fact they're subject to degradation. So in this case, hyperphosphorylation of IP3 leads to its degradation and termination of the signal that way. Any of our pathways that involve these intracellular and extracellular concentration gradients, like the calcium gradient and the sodium-potassium gradient, are going to rely on active transport by our pumps, restoring those cytoplasmic calcium, sodium, and potassium levels. Right, so lots of different ways for termination of the signal to take place. And as we said before, that's really important to maintain responsiveness of the cell to additional stimuli. You can't just stimulate a cell once and then have that cell no longer able to continue to receive information about the outside world, right? You have to be able to terminate. All right, so the last thing I want to talk about is this final mechanism of termination, which is receptor internalization. And this happens through a process called receptor-mediated endocytosis. So we talked about endocytosis previously in the course. That's just where the plasma membrane essentially engulfs something and takes it into the cell. In this case, it's just the plasma membrane itself that's coming into the cell in the form of a vesicle. And this is happening in the context of cellular signaling. So up here in blue, we have the plasma membrane. We have all these little red receptors here, and then these little blue balls are meant to represent ligand binding to the receptors. So when ligand binds to receptor, obviously we have some sort of intracellular signal transduction pathway that's being stimulated. And oftentimes that's being mediated by these proteins that are up here near the membrane. And so one way that we can shut off this signal is to actually take these receptors with the ligand bound that are all sending their signal to the cell, right, and remove them from the cell surface. And in doing so, that by itself is going to shut off the signal, right? So this is what that would look like. So you'd have an endocytosis event where in the region of the membrane where we have a cluster of these activated receptors with ligand bound, we would see an invagination of the membrane, pulling that region of membrane in, ultimately pulling it in the form of a vesicle, right, which is then internalized into the cell. And you can see here we've still got our receptors, we've still got our ligands, and these are referred to as early endosomes, right? They're sort of related to lysosomes. You might think of them as an early lysosome, an immature lysosome. And what happens after they are internalized into the cell is that they undergo a sort of a maturation process that makes them look a bit more like lysosomes, right? And so that might be something like acidification of the internal environment here within the endosome, so a decrease in the pH. And what that is going to trigger is a dissociation of these ligands from the receptor. So the ligands are going to kind of pop off here, right? And so the ligands are now no longer bound to receptor, and then what we see is a sorting event where the membrane receptors are sort of pulled together in one region of the membrane and sort of break off as their own vesicle and traffic back to the plasma membrane without ligand bound anymore. And then what remains, the vesicle that remains, is what contains the ligand that has unbound from the receptor. And that's targeted to the lysosome for degradation. So hydrolases will degrade those into their component monomers, and those could be recycled for use in the cell for whatever the cell needs to do. But these receptors have now been returned to the cell surface. They are no longer sending their signal, and they're ready to receive new signal once again. All right, so this is receptor internalization through endocytosis. And it turns out that this receptor internalization is actually mediated by a set of proteins called clathrin proteins, which coat the interior surface and assemble on the inner surface here of the plasma membrane when we have a collection of these receptors that have been activated by ligand. And these clathrin proteins self-assemble in a very elegant and characteristic way to form a sphere. And so when they start binding to one another, that's what actually pulls the membrane into this spherical shape and causes the formation of a vesicle. So this is referred to as a clathrin coated pit, this little invagination of the membrane. And this is what that clathrin protein looks like. It has this very characteristic three-armed structure that's referred to as a triskelion. and each clathrin protein can associate with other clathrin proteins and self-assemble to form this almost looks like a soccer ball structure, and these are those vesicles that are enclosing those receptor internalization vesicles. So it's a pretty cool mechanism, and in lecture I will actually show you a video of what that internalization looks like. We'll look at that video. I've also posted it to the Blackboard folder so you can look at it yourself. It's an amazing example of spontaneous molecular assembly, which happens a lot. This is just one specific example of how it's used to terminate a signal in the context of cellular communication. So that's it for our discussion of cellular communication. We've talked about its importance in a biological context. We've talked about how diverse the signals are that cells can respond to. We've looked at the main types of receptors that are used to transduce signals from the outside world to the inside world, and we've seen examples of second messengers that are used to transduce signals from the receptor to the interior of the cell. We've seen specific nuclear and cytoplasmic responses that are elicited, and we've talked a bit about the importance of a cell being able to terminate the signal in order to maintain its responsiveness in the context of a dynamic world and dynamic environment, and a need to dynamically respond to those changes.